Sunrise inside the hive, and the temperature at the center is already locked between 33 degrees Celsius and 36 degrees Celsius, and it will stay there all day. There is no thermostat. There is a living climate control system, distributed across thousands of bodies, and it is about to switch on. The engine of that system is a pair of wings most people associate with flight. During hovering flight, those wings beat roughly 230 times per second, with wide amplitude, producing aerodynamic lift. By 7 in the morning, as the sun starts heating the hive from outside, a group of workers takes position at the entrance and switches the pattern. Frequency drops to around 174 beats per second. The angle changes, the amplitude changes. Researchers publishing in the Journal of Experimental Biology in 2017 described this behavior as wings as impellers, wings functioning as propellers, not flight surfaces. High-speed video confirms the distinction is not subtle. Fanning is a separate kinematic signature from hovering, and separate again from the scent fanning bees use to disperse pheromone. This is not miniature flight happening indoors. It is the same motor, running different software. The bee did not build a fan. It requisitioned an organ built for aerodynamic lift and turned it into an industrial ventilation turbine, without swapping a single part. The workforce assigned to this task is not random. Colonies show broad inter-individual variation in the temperature threshold that triggers fanning. Some workers switch on early, some hold out longer, a spread researchers attribute in part to the colony's own genetic diversity. Field measurements at hive entrances show fanner density, air velocity, and air temperature are spatially coupled. Wherever the air is hottest, fanner density rises to match it, in real time, without any single bee directing traffic. The correlation holds across entrances of different widths and across separate colonies monitored over multiple days. Nobody assigns positions. The positions simply appear, correlated with the physics of the problem. The system, though, does not cool with air alone. Around 9 in the morning, a second group activates, the water carriers. They leave the hive, collect droplets from nearby sources, and return to distribute them across internal walls and combs. Not pooled, but spread thin, positioned with precision. Other workers direct airflow across those wetted surfaces. The water evaporates. Heat leaves with it. Research on the physics of this process is explicit on one point. Bulk water pooled in a corner does almost nothing. Cooling scales with the surface area of wetted comb exposed to moving air, which is why the droplets are placed, not dumped. A single worker exposed to high temperature individually will regurgitate a droplet of nectar onto her own proboscis. Evaporation on the tongue cools the head, and the resulting gradient pulls heat out of the thorax. This is liquid cooling operating at 2 scales simultaneously, the colonies and the individual bodies. The demand for water itself is regulated the same way nectar foraging is. When internal temperature climbs, recruitment of water carriers increases. A control loop documented as far back as 1997 in a foundational study on how colonies manage their water supply. By noon, that same airflow is doing something else, without anyone having reprogrammed anything. Nectar, brought in by foragers, arrives with up to 80% water content, too unstable to become honey. Before it gets sealed under wax, it needs to drop to 18% moisture or less. The air cooling the brood is the same air drying the nectar in the open cells right next to it. The hive does not have an air conditioner and a food dehydrator. It has one apparatus doing both jobs, at the same time, in the same space, with no individual bee, aware that it is participating in 2 industrial processes at once. Carbon dioxide inside the hive follows its own daily rhythm through all of this, rising and falling on a roughly 24-hour cycle even in colonies with no brood present at all, tracked continuously enough that researchers now propose CO2 monitoring as an early warning signal for colony health. By 2 in the afternoon, with the sun at its peak, ventilation alone is no longer enough. Older workers, past roughly 2 days of age, take on the bulk of the heat-generating and heat-managing labor, while younger bees hold position in warmer zones of the nest, a division of labor by age that colonies maintain without any supervisor assigning shifts. A new behavior kicks in here as well. Workers position themselves directly over the hottest interior sections of the hive wall, absorbing heat with their own bodies. Researchers called this heat shielding. The honeycomb receives shielders. The brood comb receives more. No bee in the colony has access to a map comparing the 2 combs. And yet the priority shows up, consistently, measured across experiments. When that is not enough, a portion of the population relocates outside the entrance, forming a visible cluster, bearding. Every body removed from the interior is one less source of heat inside a sealed space. It is thermal load reduction by physical evacuation, decided without any identifiable central coordination. Under sustained heat stress, the physiology of individual workers shifts too. Vitellogenin levels rise, a change associated with greater resistance to oxidative stress, while viral loads inside the colony tend to fall, a side effect nobody engineered, observed simply as a byproduct of the heat response itself. Up to this point, the system solves exactly one problem: keeping the hive cool. But the same mechanism has a second mode, and it does not appear every day. When a giant Asian hornet invades territory held by colonies of Apis cerana japonica, the workers' stingers cannot pierce the invader's exoskeleton. Instead of stinging, they surround it. Hundreds of bees form a dense sphere around the enemy and activate the same muscular engine used for fanning, except now it generates no airflow at all, only heat. Within 5 minutes, the temperature at the center of the ball reaches 46 degrees Celsius. CO2 concentration climbs to 4%. Relative humidity passes 90%. The hornet dies within 10 minutes. The cold detail: in normal air, that same insect survives 47 degrees Celsius for a full 10 minutes on its own. Heat alone does not kill it. It is the combination with elevated CO2 that lowers the threshold required. The bees are not simply cooking the predator, they are also slowly suffocating it in the process. No sting apparatus is ever found on the hornets recovered from these balls. The kill is entirely thermal and atmospheric. Related hornet species respond to the same tactic differently, which is its own quiet data point. European hornets die at a lethal threshold of roughly 44 degrees Celsius, yet the heat balls formed against them by other bee subspecies average closer to 40 degrees Celsius, a temperature that, on paper, should not be lethal at all, and yet often is, once carbon dioxide is factored in. The safety margin in this process is narrow. Lethal temperature for the hornet sits between 44 degrees and 46 degrees Celsius. Lethal temperature for the bees themselves is only a few degrees away, the same system that keeps the brood at 34 degrees of its own destruction, deliberately, by the collective choice of hundreds of individuals that never communicated about the plan. Genetic studies published in BMC Ecology and Evolution identified the protein rhodopsin as a candidate internal heat sensor during this behavior. Researchers believe it plays a role in how each bee tracks its own body temperature inside the ball. The complete signaling cascade, however, has not been mapped. Science does not yet have an answer for exactly how each individual worker knows when to stop. What the data do show clearly is the cost. Bees that participate in the heat ball have dramatically shortened life expectancy compared to those that do not. Even among individuals with elevated expression of heat shock protein genes, specifically built to withstand this kind of exposure. The system works. The hornet dies in under 10 minutes, in most recorded cases. The participants in the ball pay the price in measurable, documented, repeatedly replicated weeks of lost life. Natural bee balls, researchers note, are typically sustained for 20 minutes or longer, well past the point required to guarantee the kill, and well past the point of diminishing returns for the bees still vibrating inside it. By the end of the day, the same set of muscles that cooled the brood in the morning, dried the nectar at noon, and formed a biological furnace in the afternoon, returns to default mode. Simple ventilation, controlled evaporation, temperature held steady between 33 degrees Celsius and 36 degrees Celsius. There is no record of any single bee ever noticing that it spent the day operating 4 different systems with the same equipment. The colony, in the end, never knew it had an air conditioner. It only knew it needed moving air, and it used what it already had.